for our first session, I'm honored to introduce the path-breaking scientist, Dr. Jennifer Doudna. Jennifer is a foreign member of the Royal Society. She won the Nobel Prize in Chemistry in 2020 for her co-discovery of CRISPR. She is the president and founder of the Innovative Genomics Institute and a co-founder and advisor to several companies developing unique CRISPR-based solutions to tackle some of the greatest challenges in health, agriculture, and climate. She is considered one of the foremost and most pioneering influential scientists of our time. Please help me in welcoming Jennifer to the stage. Thank you. Welcome.
James, great to be here.
Good to have you. Now, Jennifer, it's been, 4 years since you won the Nobel Prize in chemistry. And since then, a lot has happened. I'm curious, would you share with us what's top of mind for you, both in your research, but also things going on in your field?
Well, it's amazing to be here. What an incredible opportunity to visit with other scientists and hear perspectives on AI and science, something we think about a lot in my own research and at the Innovative Genomics Institute. I think with CRISPR, which is a technology for editing DNA precisely in cells and organisms, what we're seeing is a real step change in the pace at which this technology is being applied. And one great example of this is the approval of the first CRISPR therapeutic to treat patients with sickle cell disease. It was actually approved here in the UK first, about a year ago, and shortly after that in the US. And it's a functional cure for a human genetic disease. It means that patients with this terrible disease can receive a one-and-done treatment that deals with their disease at the genetic level. It makes a change to DNA that overrides the sickle cell mutation and means that these patients at least so far, have been symptom-free since receiving the therapy. It's extraordinary.
That's
Several
incredible.
dozen people have now been treated. And so it's for everybody in the field, it's been a tremendously exciting moment and really illustrates the partnership between scientists, of course, patients that were voluntary participants in clinical trials, and then, of course, the companies that have been making all of this possible. So I think it's a reminder of why we do the science that we do and the extraordinary opportunities that lie ahead.
No, it's incredible to see how you go from the science to breakthroughs to actual things that start to make a difference and drugs that start to make a difference for people. Now, obviously, how AI fits into this picture is top of mind for many of us. What do you see as the key opportunity areas for AI within gene editing and CRISPR, but also just in genomics research more generally?
Well, one of the things with CRISPR, and thinking about this approval, this drug approval, is how we expand the applicability to people around the world. Right now, this therapy costs about $2 million US per patient, so it's very expensive, meaning that it's difficult to get a hold of financially, but for many people, but also just the way it's applied, it requires a bone marrow transplant to receive this therapy. So we see a future where we override that barrier by discovering better ways to deliver CRISPR enzymes, making CRISPR enzymes that are more efficient at what they do. So you need to use fewer molecules to achieve a clinical benefit. And then, of course, expanding the utility of this gene editing platform in other areas. And all of those are spaces where AI will play a big role, I feel. And also, it should be mentioned that CRISPR, in addition to being a tool that is directly applicable clinically and also in the environment, I'm sure we'll talk about that, it's also an extraordinary research tool. So it generates a lot of data that are then suitable for mining by AI-mediated approaches.
So does that, in the end, also accelerate the research pipeline? Does it mean that we'll get drug therapies and other things faster?
I think no question. That's not happened yet, but I think many of us see that that's on the horizon.
Yeah. When most of us think about gene editing, we tend to think of medical therapies as the arena where this will play out and benefit. But it's actually much broader, isn't it? I mean, at your institute, you're starting to work on other areas like climate change and other things. Say more about that. What do you see as the kind of the broad applications of gene editing?
Well, when we founded the Innovative Genomics Institute about 10 years ago, our thinking was to focus in both healthcare and climate. These are two areas where we think gene editing is going to have a major impact. And people don't necessarily think about genome editing and CRISPR when they think about climate change. But we see incredible opportunities both to make changes in plants that do things like allow better carbon storage in the soil through root systems, and to create drought-resistant plants like rice, which is something we're doing at the Innovative Genomics Institute, and also, frankly, to use CRISPR at its source, namely in the microbes that populate our environment and our bodies, and that impact our health and, of course, commercial agriculture.
Well, you're going to have to tell us about the cow that Hannah
Oh, yeah,
mentioned.
the cow, right? Yeah. So we were talking at dinner last night about where many of us see the immediate or, say in the next 5 years, impact of AI in science. And my answer was a methane-free cow. It turns out that cattle are one of the major sources of methane, which is a powerful greenhouse gas that's emitted through, agricultural practices around the world. Imagine that we could create cows that were literally not burping out methane and therefore using their feed more productively. Farmers would like this. And of course, the rest of us would like it. And so this is something that we're working on at the Innovative Genomics Institute in partnership with University of California, Davis scientists who have demonstrated that in principle, this is absolutely possible. They do it today with complicated manipulations of the cow diet. We see a future where genome editing could be used in the cow microbiome, which are the microbes that populate the cow rumen, which are actually producing methane, and change their metabolism so that no longer is a problem.
Oh, wow. Oh, wow. Well, it's extraordinary to hear you talk about this and how gene editing has kind of gone from the stuff of science fiction to actually being, kind of an established tool and protocol for how we do a lot of other things, and also how you and your colleagues are navigating through all of this. Well, here perhaps is a provocative question, if I may. Do you think advances in AI make certain elements of gene editing obsolete?
I actually think that there's an incredible opportunity for a collaborative approach. I think that AI advances our capabilities as scientists, or it can. And what I see is a future where we can more accurately predict the outcomes of experiments. For example, genome editing experiments. We know which genes need editing and which ones are superfluous to an outcome that's desired. Even predicting things like the impact of editing over time in an organism. And I think that will actually accelerate the pace of discovery even faster than it is now. And we hope make some experimentation unnecessary. So I think that would actually be an incredible advance if that comes to pass.
So it might even help kind of steer or suggest or guide what experiments to do and which ones not to do and which ones to explore and so forth. That's extraordinary.
Exactly.
One of the things you've made observations about with CRISPR, and we're also starting to see in AI, is the resource-intensive nature of the research itself, which is also shifting kind of traditional relationships between academia and the private sector, and also changing how we think about funding. How do you assess kind of the current funding landscape, and what can we do to change to facilitate the kind of research like CRISPR or AlphaFold level breakthroughs happen even faster and perhaps even more collaboratively?
Well, James, I've been an academic for a long time. I've been running an academic lab 30 years. And I'm also now more recently a founder of companies and have worked a lot with new company teams that are building groups that are focused on particular applications of CRISPR. And what I see is that I think there's an incredible opportunity that hasn't quite been advanced yet, and in which we, Innovative Genomics Institute, would like to be at the forefront of, which is to think about how we fund innovative early stage science to enable would-be entrepreneurs to make the kinds of creative discoveries that will then be the foundations of new companies. And frankly, it can't, at least in the US, I don't know how it is here in the UK, but I imagine you face similar challenges. It can be very difficult to get government funding for very early stage, high risk, but potentially very high payoff ideas, especially with young scientists who are, just starting their careers. And that's really what we aim to do at Innovative Genomics Institute, is to fund those scientists. And we do it in a way that enables them to work collaboratively, which is often not necessarily rewarded in academic settings. We actually reward that. We ask them to work together in teams, and we ask them to consider interdisciplinary approaches to problems. And so, for example, our microbiome work with the cow is very much that way, where we have computer scientists, even electrical engineers, and agricultural scientists who are all working together. These are normally, at least in academics, not people who would necessarily bump into each other.
Right.
But we build the teams that make them possible.
No, I have to say, it's really quite inspiring, Jennifer, to hear you continue to do this extraordinary path-breaking scientific work, but also sounds like you're starting to innovate how we do science and create ventures and structures in order to do science. I have to ask you this, though, which is, you have a unique point of view as somebody who has been in the eye of the storm, if you like, with regards to a research breakthrough that people on the one end find hugely exciting and sometimes also find quite concerning with societal implications. What can all of us working on AI learn from your experience with CRISPR? Because you could argue AI has some of the same exciting and complicated and concerning characteristics.
Absolutely. Well, I think back to 2000, I think it was 2014, the end of 2014, we had published our work on CRISPR in 2012. And by the end of 2014, scientists were already using CRISPR in monkey embryos that were implanted and were genome edited not just in particular tissues, but in the entire organism, such that they could pass those gene edits to future generations. Right. And so when I read that academic paper, it was very clear that I didn't see any reason why it wouldn't be possible to do it in human beings. And as some of you here may know, that in fact did come to pass. Right. And so what that motivated for me was the realization that it wasn't going to be enough just to put my head down and work in the lab or work with our institute. We really had to be implementing a whole team, and we have this now at IGI, that works on public impact. And we ended up involving the Royal Society and the national academies in the United States, as well as equivalent organizations in other countries, to boot up a series of international meetings on human genome editing. And so, in fact, the most recent of those was held here in London in March of last year. And that's led to a very open, transparent conversation. It's gotten groups involved, including the World Health Organization, to encourage a transparent database of work that's going on using genome editing by scientists, especially if they're using it in the human germline, or for applications in the environment that could also have global impact and could be dangerous if not used properly. So I think one thing that we've learned from that experience is that it's incredibly important to encourage that kind of open, transparent conversation. It can't just involve scientists, it has to include other stakeholders. We work very hard at Innovative Genomics Institute to make knowledge and data accessible to people that are not experts, right? So we have a lot of high school teachers, for example, that we work with, that we give them resources to help educate their students about genome editing, what it means and where it's going. So I think this is something that will be incredibly valuable also in the AI space, doing the same kind of encouragement of public discussion, knowledge-based decision-making. I mean, this is just so important, especially now.
Absolutely, and we're trying to learn from your examples and the paths and approaches you've taken. I mean, in much of the work that we do, including in AlphaFold, I mean, my colleagues Demis and John and others went through an extraordinary process with ethicists and others before that work ever saw the light of day, to think through what are the implications and possibilities of all of that. But there's so much more still to learn. Indeed. But I wanted just to thank you for taking the time. There's so much to be excited about here, and it's an absolute honor to have been in conversation with you. And thank you for all the work you're doing.
Thank you. Thank you for including me, James. Great to be here.